the overview, the branches of the microbiology. So I'm not sure you guys are familiar with the fungi or parasite, but I'm sure you know about the bacteria, because bacteria is the most common one. It's the E. coli, Staph aureus, like Staph pneumonia. You probably heard a lot about E. coli, right? E. coli, and then virus. I'm pretty sure you guys know about this very well during the COVID-19, because COVID-19 is one of the virus. So when we talk about microbiology, it's a study about the microorganism, which are microscopic, that include the bacteria, virus, and also the fungi. So all the four components. Okay, right. So but for my lecture today, I'll focusing on the virus. So when we talk about virus, this is how it looks like. Okay, on the left side, it's something that really beautiful. Okay, and on the other side, on the right side, it looks like something that very painful and nasty. So the key point here is that both of this condition is actually due to the virus infection. On the left side, it is due to the um, pattern mosaic virus, which is causing the stripping patterns on the tulip flower. And on the right side here is actually due to the smallpox. So you can see here, it shows that virus not only can infect human, but it also can infect animal. It can infect plants, and the effect is actually different based on where it actually infect. For example, for the flower, it caused something beautiful, but then for in human, it caused a disease. So it's a wide range of effect. Okay, so the size of the virus, if you look here, it's very small. At the bottom here, the figure here, you compare between the virus and E. coli. If you look at the E. coli, it's very big, right? And if you look at for the virus, for example, like adenovirus, okay, the respiratory one, it's only 90 nanometer. It's very small. And even if you look at this figure here, they put the herpes virus, okay, on the surface of the cell. It's very tiny. Okay, you cannot see them with the naked eyes. You have to use the microscope electron to see the structure. Okay, herpes virus, even here, one of the biggest virus, but again, it's very small if you compare with the bacteria. Okay, so this is the structure of the component, the structure of the virus. Sorry. So the structure of the virus. When I say virion is actually is a mature infectious particle that can infect host cell. Okay, can infect a host cell, and the capsid is actually the protein that enclose the nucleic acid. Nucleic acid, it can be the DNA or RNA. So the stuff that cover that nucleic acid we call as a capsid, and this capsid can be found in different form. The blue color here, this one is in a helical capsid form. This one is the icosahedral, and this one is the complex form. So, and then they also have capsomere, and they have nucleocapsid, and also envelope. Envelope is the outer layer. So outer layer is very important because this one, on the surface of the envelope, you will see the spike protein. The spike protein is very important for the attachment process. Okay, and then some virus, they exist without the envelope, and that one we call it as a naked virus. Okay. All right. So when this is the simple classification of the virus, okay. So virus can be divided into two, whether it is DNA or RNA, but it cannot be both. So they have to choose only one, either RNA or DNA. So for DNA, you can see it is divided into um 8 groups here. Okay, 8 group, you can see they have different types. So first thing they look for is the nucleic acid, and then the next criteria is we look at the capsid, whether it is symmetry or not, and then whether they are naked or envelope, and then further they will divide based on the genomic material, whether it is single-stranded, double-stranded, linear, or circular. Okay, and then for RNA, this is how it looks like. They have almost like I think 14 groups of RNA virus. So RNA virus, second criteria will be looking at the capsid, and then they look whether they are naked or enveloped. And then after that, the genomic architecture, whether it's double-stranded or single-stranded. Okay, all right. Okay, so the replication cycle, how virus infect the cell. Okay. Um, is it okay if I ask question? Are the students willing to answer? Yeah or no? Anyone interested to answer if I ask question? Do you guys know if a virus is actually a living or non-living? Living or non-living? Anyone? If you're too shy, maybe you can write down in the chat. No? Okay, so actually virus is actually, we call them is a, it's a living, but indirectly it is in living. Why in living? Because the virus itself cannot multiply by themselves. So they need to use the whole cell machinery to replicate. So it means that without the whole cell, they cannot multiply. If they exist as one one single virus, it's going to be one single virus forever until they found a suitable host cell. So once they found a suitable host cell, they will go through the replication cycle. They borrow the host cell machinery to replicate and then to produce more virus. Okay. So basic steps in virus replication. First one is the attachment of the virus. So attachment will be the binding between the spike protein on the virus and also the receptor. Remember, I told you on the surface of an envelope there will be a spike protein, right? So the spike protein will bind to the receptor on the host cell. Okay. After binding, you proceed with the entry. So entry means the cell will penetrate, and then uncoat. Uncoat means that if they have an envelope, they will open the envelope to make sure that they can release the genomic material to go inside the nucleus and then proceed with the replication and also the gene expression. So the pink color here is actually the gene, the copies of genomic, whether it is DNA or RNA. It's up to the virus itself, the origin. And then the blue color here is the viral protein. So this viral protein is necessary for the during the packaging. Okay, so packaging is like when you produce something, when you produce a product, at the end of the day, you have to pack them, right, before you deliver. So the process of packaging, we call it as assembly. So assembly is like putting the genome, putting the viral protein, and what creates the membranes, create the envelope, and then it becomes a new progeny, a new virus again. So the last one will be the maturation and release. So this maturation was, there are two types of maturation process. Certain virus will go through the maturation before the releasing, and that, but then some virus will will go through the maturation process after they release. So it's either before release or after release. So this one is depends on different types of virus. Okay, this is the virus goal. So now you know how the process of the process of the replication, right? So let's start with the attachment, and then penetration, okay, the entry, and then the synthesis. Okay, so this is how it looks like when we plot them, when we do the PFU, the plug assay. So this is how it looks like. Okay, so the first thing here, you see the latent period. So it's like a mendata. So it's a latent period here. So during this latent period, what will happen is that there will be a virus attachment, virus entry, and also the synthesis. And then after that, once the virus is ready, finish with the assembly, and they will release the virus. So that's why you see the peak, the increasement of the peak during, that's what we call as a first burst. So first burst means that the virus is released from the infected cell. Okay, and then this new virus or variant, because variant is the one that infectious, right? So this variant will be released out again and then infect new cell. So once they infect a new cell, so that's why you see another latent period here. So this latent period is actually the process of attaching, entry, and also synthesis. And then they produce more virus. So that's how you see the second burst. So this one is will be repeating again and again until the dose is good enough to cause the disease. So basically, this is how it looks like. Okay. So first burst means that a new virus is released and then go infect other cell and then release again. So that's why you see multiple peak of virus burst. Okay. All right. So virus host range. So we know that the virus needs to infect cell to replicate, right? But how they choose which cell is suitable for them? So this one is depends on this spectrum. Some virus they have a wide like a broad spectrum means that they can infect more than one species. For example, the swine flu, which is the influenza, it's infect everything, almost everything. Human, chicken, tiger, cattle, like swine. So it's infect multiple species. So this one we call it as a broad spectrum, broad range, host range. Okay. And then sometimes there are also certain types of virus that only infect one species, which is human. For example, the smallpox virus. Okay. And then sometimes also it's not only specific to one virus, but it's specifically only to one tissue type or type. For example, HIV. HIV is very specific because it's only infect the white blood cell, and specifically the CD4 T cell. That is the most favorite one. So they attack this one they attack this CD4 T cell to replicate inside them. Okay, so that's why CD4 is one of the indicator for HIV disease progressing. So if you see the CD4 cells is dropped, so means that the prognosis for that patient is very poor. Okay, so sometimes, like I said, there are certain types of virus very specific. They have a even specific T cells that they want, which is the CD4. Okay, and in some virus also can infect different types of tissue. For example, adenovirus. Adenovirus, even though it is classified under respiratory, but it also can infect eyes, which is it can cause the conjunctivitis, and it also can infect the gastrointestinal tract. So this one also can cause the diarrhea. All right. Okay. So now you know about the host. Okay. But then how come, or what kind of criteria, what kind of factors that actually um, influence whether that individual is getting a disease or not? Okay, because for example, you during the COVID-19, okay, so everyone's living in the family, okay, some of them positive with the COVID-19, but with no symptoms, and some of them with a very bad symptoms, and then even sometimes one individual in that family did not get the infection at all. Okay, so why is this happening? So these are the factors actually, the factors that influence whether that individual is actually getting the disease or not. Okay, so there are two different factors, the host factor and also the virus factor. So virus factor it depends on the virus. Okay, the first one is the dose, whether the dose is good enough to cause the disease or not. Okay, and then the route of entry. Route of entry means that whether it is specific. For example, like adenovirus, if it goes to the respiratory tract, it can cause the pneumonia. If it goes to the through eyes, it can cause the conjunctivitis. So it depends which entry. Okay, and in tissue tropism, this one that I just explained just now. Okay, well, sometimes they have a broad spectrum, sometimes very specific. Okay, and then virus genotype, virus virulence genes. This one, if you think about COVID-19, remember during COVID-19, we have a different kind of like strains, like a new strain will come out. So this one is also affecting whether that person is getting disease or not. For example, you already infected with COVID-19 before. Supposedly, if you are already infected, you are protected, right? So it means that you will not getting the disease anymore if you get it for the second time. But due to the virus genotyping, because they have changed, they have gone through the mutation. So when you get it for the second time, the symptoms might get worse. So this is the reason. Okay. All right. So host factor, this one is actually related, highly associated with the immune system. The key point here is the immune system. Like for age, your age right now is the best, the perfect one, because you guys are at young age, okay, still energetic, so your immune system is well developed. But for those who are younger, like for kids, okay, for babies, they are not well developed, so they are risky to get infections, okay. And then after that, for the elder people, the immune system is probably already weak. Okay, they reduce the production of the immune cell, like a B cell and also T cell. Okay, that is why um, the older people can get infected easily. Okay, then host genotype. Sometimes this one is more like genetic disorder. Some people, they are susceptible to certain kind of infections. So you have to be very careful if you have this genetic disease. So make sure you please avoid any kind of like possible transmission to get the infection. Okay, nutritional status. Of course, if you eat healthy, okay, you will get good nutrients, so the your immune system will be good. And then other infection. Other infection means here that if you're getting a infection, for example, like bacteria infection, and you have to take the antibiotic, okay, for treatment, but then at the same time, that antibiotic causing your immune system to suppress. So when immune system is suppressed, it's easy for you to get infection with other pathogen at the same time. Okay, so that's how it is related. So this is an example of how the host factor plays role during the COVID-19. Of course, so during the COVID-19, most people died not because of the virus directly, but they actually died because of the activation of the immune system. Okay, they say that the cytokine storm that happened during the COVID-19 infection that caused the disability to the important organs in humans. So that's why there are a large number of death cases for COVID-19. Okay, and then for comorbidities, there are the factors with other diseases, especially for older people. Okay, those with the diabetic, obesity, hypertension, they are more risky to get COVID-19. Okay, genetic factors. This is the one that I told you. Okay, some people, they are special. They have the genetic disorder, and then certain types of genes are more susceptible to COVID-19. And then microbiome, this one is co-infection. If you have infection with the other virus, with other bacteria, you take antibiotic, and then you also might get the co-infection with the virus, right? Okay, this one is ecological factor. This one is more on involve the environment. If you look at the diagram, it shows here that it's a migration of birds. So usually, let's take example for influenza. So influenza virus can be transmitted through birds, migratory birds, to chicken, right, to through animal. So this movement population actually give a high risk to transfer, and then to develop a new outbreaks for infectious diseases, especially for virus. Okay, and another one is for this one is uncontrolled urbanization. This one is more related with the vector associated diseases. For example, dengue. Like the mosquito. For example, like here it says that the breeding in cans, old tires, and so this one will give like a place for the production of the mosquito, because mosquito will have the virus, right? So mosquito will bite human, and that's how human get the dengue virus from the mosquito. Okay, and then for the, yeah, this one is long distance livestock, means that the transportation by the wild birds. So they move to another place, and then they go infect um, the local birds, means like a chicken, at the farm, and then that's how they get infection to the farmer. All right, so this is another example of the COVID-19 um, ecological factor or other factors are involved in COVID-19. Okay, for example, like mobility and transportation, of course, the geographic, this one is more related to the genetic, because sometimes those who are in UK are more susceptible to certain strains of COVID-19, right? So that's how it is due. And then chemical and microplastic, for example, this one is microplastic can be a carrier for the virus, because virus can stay, okay, at the surface of the plastic, and then it can become infectious when other people touch it. Okay, and in nutrition, of course, this one is related with the immune status. Okay, so this is how, talking about the transmission. So you know about the how it is the factor that involve. So now we are talking about transmission, how it is transmitted. So the routes of transmission, there are two types. One is the horizontal and one is vertical. Horizontal means that something that really quick. For example, like COVID-19, influenza, okay, where COVID-19, Ebola virus also can spread through the horizontal, means that from one person to another. And the medium can be like water, food, or direct contact. Okay, and the vertical is something that you pass from one generation to other. Like for example, this one is from a mother to the baby, and it can be like HIV, or maybe like rubella. So this one is we call it as a vertical. So horizontal versus vertical. And then for the horizontal, there are different types of modes of transmission. Okay, if it involves the respiratory, okay, we call it as respiratory. Fecal oral spread means this one is, for example, like you have digested food, water that contaminated with the feces that has the virus. Okay, venereal spread. This one is probably due to the sexual activity. Okay, and then a vector. Okay, this one, if it's involved vector or reservoir animal, we call it as a zoonosis. Okay, so vector is like a transporter. So this vector will transport the virus and infect from one person to another. Okay, and a reservoir is actually referred as a tank. That is where the first stop of the virus comes from. So it's involved two things here: reservoir or vector. Okay, I'm going to give you an example here. So about the COVID-19. Okay, so COVID-19. This one is not the COVID-19, but it's a coronavirus. So before COVID-19 came, okay, in 2002, there were SARS-CoV-2. So SARS-CoV-2 came in 2002, and then MERS-CoV came in 2012. So the duration is almost 10 years. And then after maybe, I think 8 years after that, the COVID-19 came. Okay, so be prepared. It might come back again in 10 years, we never know. So how and why did they come back? Please go back to the previous notes and how it's involved the host and also the virulence factor. So based on that factor, maybe the new virus will come back again in 2030. Okay, this is just a prediction. So based on this, okay, so it's involved more than one animal. So if it's involved more than one animal, one must be the um, reservoir, one must be the vector. Okay, so intermediate host here is a vector. Okay, the bat here is the reservoir. Okay, how it happens is, well, let's talk about the SARS-CoV-2. SARS-CoV-2 happened in 2002. This one also started in China. Okay, what happened was that the bat, so the bat is actually is the reservoir. So the virus is there inside the bat. So the bat eats the fruit. Okay, and then the leftover. So the bat just leave the leftover, and then the leftover was eaten by the intermediate host, which is the palm civet. Okay, so this palm civet, okay, the farmer catch the palm civet and sell them at the market, right? So this palm civet is a vector. So sell them at the market. So at the market, that is how the palm civet spread to the human. It's probably by touching the saliva or maybe by sneezing, because it's the respiratory virus, right? So anything from the nasal or from the mouth can infect other people. So from that, the human will affect other people and then spread. So this is how the horizontal um, what horizontal transmission happen. Okay, same with the MERS-CoV. MERS-CoV, it's the same coronavirus, but different pattern because it's involved a camel and it's happened in the Middle East only. Okay, and it's they say it started from the bat too. All right, so for COVID-19, this is the horizontal transmission. There are two ways. One is direct, one is indirect. So direct, of course, from direct person, from the aerosol itself. From for indirect, it's probably because of the droplet of the sneezing or coughing. So that droplet will fall at the any surfaces, okay, any surfaces object, and then the individual contact that surface and object. So that's how they carry the virus, okay. So and then it can spread to other community and also social contact, even in the house or like in the office area, okay. So that's the spread. So different types of virus production. Okay, when you talk about virus, it does not means that the pattern of infection is always the same. No, because some virus you can solve the problem like very quick, but sometimes when you get the infection, it stays there inside your body for the rest of your life. So that's why it's very important to do the screening. Sometimes you don't know you get the virus infection until it already at the end of the stage. Okay, for example, like this one, persistent infection, LCMV. Okay, LCMV, it's persistent infection means that once you get the infection, you get it for the rest of your life and until it causes death. And this one can be carried out from one generation to another. You get infection, you don't know if you don't know you're positive with that virus, but then you have kids and there's something wrong with your kids, and usually will this one will cause a congenital defect. That is how you know, oh, I actually got the LCMV infection. So and this one is herpes. So herpes simplex virus, it's very common. There are two types. The one that causes the blister at the lips, and the one that causes the disease at the genital. So the one that causes blister at the lips, sometimes students say, they're not aware of it, but it's actually it's due to HSV. So you also need to check on this. And then for HSV, once you have it, you will have it for the rest of your life. Okay, you cannot resolve it. It's there. So that's why it's very important to take care of your immune system so that you don't get infection with this kind of virus. Okay. And then last one, the slow version. This one is HIV. HIV, the first peak, here it shows the symptoms, right? So first blue peak is the symptom. So you get the infection, usually the first symptoms will be like flu-like symptom. You just get like fever, headache, like stomach ache and everything. So it's a common one. So you don't know, and then the virus will stay inside your body for 20 to 30 years. So at the end of the day, the virus will come again, not come, but it's still there, activated, get activated again, and that you will get the co-infection. So during this time, you probably get the infection with the fungus, like candidiasis, infection with the parasite. So you have multiple infection, and then after that, you can just can cause death, right? So this is the patterns of infection. The one that easily can resolve is actually the first one. So this one is the influenza. So influenza, usually you get the infection, you get flu, right? And then you got sneezing, cold, and then after a few days, maybe 1 or 2 weeks, and then it's gone. It's resolved. So that is the short version one. Okay. All right, so how it's detect? How can we detect? It's very hard to see the virus by the naked eyes. So that's why we need to use a different techniques to detect the presence of the virus. For the microscopy method, we can use it, but it's very limited because for the light emitted microscope, we need to culture the virus inside the cell, and you don't see the virus. You see the reaction of the virus based on the appearance, based on the CPE, cytopathic effect that appears on the cell site, on the cell culture. Okay, so you don't see the virus. So the green color here, this one is the light emitted microscope. The one that can be used to see the image clearly is actually the electron microscope. Okay, so this is the electron microscope. Then this one is to see the binding between antigen and antibody. So you see the fluorescent color, it shows that there's a virus based on the antigen presenting due to the binding between antibody, your sample, and also antigen, which is the virus. Okay. Quantification, we can use the blood assay, hemagglutination, and then for serology, of course, this one to detect the antibody, whether it is IgM or IgG. And in confirmation assay, this one you can use the PCR, Western blot analysis, flow cytometry, and also ELISA. I'm not sure how much you learn about this, but these are the confirmation assay, and it's more accurate if you want to check for the presence of the virus, especially for PCR, because we use a specific gene for a specific virus that we want to detect. We even can detect the strain, so that's why this one is better. Okay. So for the treatment, you can take the antiviral drug if it's available. If it's not available, you just treat the symptoms. If you have fever, so treat the fever. If you have diarrhea, treat the diarrhea. Okay, you cannot treat the virus if there's no antiviral. All right, prevention, take vaccine if available. Some vaccines are not available, so if it's not available, you have to maintain your lifestyle, like eat well, sleep well, get the exercise, okay, and then take preventive measures based on the transmission process. For example, I put here for the poster for the swine flu, okay, because it's transmitted through the aerosol. So you have to make sure that this one is almost similar with the COVID-19. So you wear a mask, okay, you keep your distance 1 or 2 meters, and then you wash your hand properly, okay, and if you have the symptoms, you go to the clinic. Okay. But for the swine flu, it put here in the red circle, you cannot get the swine influenza from eating pork. This one is only valid if you cook the pork properly. Okay, so make sure you cook them properly. If you eat them halfway or half cooked or maybe raw, I don't think it's safe. Okay, right. So now you know the overview. What is the viral infection? How it's replicate, and then what are kind of the diagnostic, what kind of the treatment. So I want to take example specific. This one is for the influenza because this is the most common one. Uh, for it always there. Like for other country, like for seasonal country like Canada and US, it's seasonal. So every year this influenza virus is there. So that's why they have to take vaccine every year. So vaccine will be given to the students every year. Okay, so influenza virus, it is highly infectious. I know you guys familiar with the H and N, right? So it's probably H1N1, H7N9, okay, H5N1. So they all are actually influenza virus. So they call HNA because of the two types of glycoprotein on the surface, which is the HA or NA. Okay. All right, so these are the chemical foundation in the structure. The protein, the lipid, the spike, the viral genome, they all consist of the chemistry of the chemistry properties. And this is very important because it determines the viral function. Okay, so that's how the chemistry parts regulates in the genomic material. Okay. Okay, so this is to show you the binding activity between the receptor and also the spike protein, also the receptor. Okay, so you can see this is hemagglutinin. Hemagglutinin is the spike protein on the surface of the virus. So hemagglutinin will bind to the salic acid. Salic acid is the receptor that exists on the host cell. So after this binding, of course this is not an easy binding. They have to go through the chemical interaction, the conformational changes. I'm not the expert of that, but I know there's a binding between the spike protein and also the receptor. So this will induce the chemical interaction, okay, between the viral entry and binding to the host receptor. So after the binding, the cell will go inside, okay, and then we'll go the entry process. So during the entry process, you can see, so here is another influence of the chemistry part where the pH is changing. pH is changing during the entry process. So once the virus bind to the spike protein, they will go inside, okay, and then they form the endosome. You can see the changes of the pH of endosome from 7 and then start to decrease. Why it has to decrease? It's because so that the virus can release the genetic material to go to the cell nucleus. Why need to go to the cell nucleus? Because they need to proceed with the DNA replication, transcription, also the translation. So for chemistry, part is that people are now looking for the treatment, right? So treatment and also like a detection. So the first part here for the receptor binding, okay, this one also can be the target for the at the chemistry level. How this can interrupt the binding between the spike protein and also the salic acid. Okay, and then this one is actually how the pH level can actually disrupt the virus, okay, from releasing the genetic material so that it can stop the virus from using the host machinery and then proceed with the replication, transcription, and then translation. So each step of the virus replication can be the target for new development of new drugs therapy for viral infection. Okay. Okay. And then also for the next process, so once the virus go to the inside the cell, okay, so they will go through the replication process. And in the replication process, we know that it's involved a lot of enzymes, for example, like DNA polymerase, and in the RNA polymerase itself, they have one and two, okay, and then RNA polymerase, which is RNA primase, DNA ligase. So this is also another step where the chemistry part of chemistry will take part, where they can target each enzyme to stop the viral replication process. Okay, and then all right, so during this replication process, there's another step called proofreading. So proofreading is like another checking process to make sure that there is the cells that producing the new cell that they produce is not going through the mutation, but somehow the proofreading activity is not very efficient. So that's why it also can lead to the mutation. So this mutation, we call it as a gene reassortment. So in virus replication, okay, okay. So in virus replication, it can cause the antigenic shift or antigenic drift. So antigenic shift, if you look at it's rainy here. I hope the internet connection is okay. Rainy heavily. Okay. For the antigenic shift, you can see here, all right? So it's involved two strains of influenza, H1, H9 and 1, and also H5 and 2. So in combination of these two, suddenly it produced a new strain, which is H5 and 9. So these new strains, okay, this is the one that will lead to the outbreak or lead to the pandemic. Okay, so let's say I give you example. You already get infected with the H9 and 1 before. Okay, you're already infected with the H1 and 9. So usually, once we get infected with a virus, our immune system will produce a memory cell. So we should recognize the same virus. Okay, but then since the new one is coming, okay, so H5 and 1 is the combination between the two strain H9N1 and H5N2. So now your poorly existing antibody is no longer protected. So that's why even though the strain is almost similar, but your antibody is not working. So that's why you still get the disease. Okay. So the chemical challenge here is that to make sure that they can develop a broad spectrum antiviral and also a rapid diagnostic. So that means that if it has been used before for the H9N1 treatment, so hopefully for the broad spectrum, it also can be used for H5N1. And then for the antigenic shift, this one is not very like it's not a huge difference. It's the same name, for example, like H9N1, but you can see there's a point mutation there. You can see the blue dot on the genomic material. Okay, even this one does not cause pandemic, but it still can cause the outbreak because the immune response that you receive during the first exposure is also cannot protect you 100% from this. So the chemical challenge here is that they have to update them regularly with the vaccine and also the antiviral. But the problem is that it's not easy to develop vaccine or antiviral, right? It takes time. So that's why I think it's important to always getting update about this information. Okay. So like I told you, for the antigenic shift, you probably like wonder how they combine between two strains. So here is the example. Okay, so the red color one is the red strain. Let's say red and blue strain, right? So red strain originally they come from the migratory birds. Okay, they come to domestic birds. So this guy, this guy he has a farm. So at the farm he has like pigs and also the chicken. So the migratory bird will come closer to the chicken and then infect the chicken. And then chicken also can infect the pig. And the red strain also can infect the human directly. And then previously, this human already have the blue strain. So what happened is that when these two strain exist in pig, so pig is a mixing vessel. We call them as a mixing vessel because pig are susceptible to any kind of species of influenza. Even though it's from human, even though it's from chicken, whatever it is, they just accept them. So they accept them, so they go through the process. So during the replication process, that is when the antigenic shift occur. So that's how they produce a new strain, which is half red and then half blue. So this is the one that caused the outbreak. So how the chemistry part will take role during the outbreak? The first one is of course through the diagnostic development. So during the outbreak, we need something that rapid, right? So very rapid, very sensitive, and of course we have to make sure that it is cheap too. So you need to develop a new method. Okay, even the PCR, you have to screen. You have to do the screening for the genes, which genes that can be detected for certain strains. Okay, and then for therapeutic innovation, of course, the chemical analysis of the proteasome will back the development of the antiviral drugs. Okay, for example, maybe we can create a protease inhibitor for SARS-CoV-2. Okay, and then another one is the vaccine development. This one is, we know that the vaccine is already there, but we also need to do something for the improvement of the vaccine that it can cover more strains and with the less side effects. Because as nowadays we know that most of the vaccine can cause the side effect, whether it is short side effect or the long-term side effect. So we have to make sure that the vaccine, the produce can deliver the mRNA, and we should recognize it as a safe mRNA, not like a foreign mRNA. If we recognize this as a foreign mRNA, so that's why there's a lot of side effects happen after the vaccination. Okay. Chemistry solving in viral challenges. So I put here 3 examples